Hi students. So when working on this determination of genotypes from Phenotypes Lab, the most important thing that I want you to focus on before you start is reading this top section. I know it seems like something you want to glaze over, but it's really important to understand that your phenotype is what you actually look like on the outside, which is often different than what your genes are, your genotype. So you might inherit certain genes from your parents, but they don't all necessarily express themselves in their phenotype. For instance, my mom had brown eyes, my dad had blue eyes, so I have the blue-eyed genes in my genetics, but I got kind of the brownie hazel eyes of my mom. So even though my genes, I have some genes for blue eyes, I have the phenotype of kind of this hazel brown-eyed person. What does that mean for me? Well, I married a blue-eyed person and together we had kids, so we actually have a few blue-eyed children, and they got that blue eye from dad and from me, even though my phenotype shows like a brown eye. So your phenotype might be different than what your genotype actually is. Following through and reading these directions are going to be really important for you, but I am going to give you a few quick pointers to go through. The first thing that you're going to do is in your lab kit you have some PTC paper. Now I pulled them out and put them in an envelope for you, so you only got one piece of the PTC paper, but it's a tiny piece of paper like this. PTC paper to me tastes really, really horrible because I am a taster and can really taste the chemical that is on this paper. It's not a dangerous chemical to you. It's just a chemical found in like broccoli, cauliflower, Brussels sprouts. And for people like me that is a taster of this chemical, this is a very strong taste, and it's one of the reasons why I had to teach myself to like those vegetables. It wasn't something I liked as a kid. So you're going to take this piece of paper to start and place it on the middle of your tongue. There's also a control piece of paper. If you didn't get the control piece of paper, you can taste test any piece of paper and taste a piece of paper and it won't have any taste probably. This PTC paper, put it in the middle of your tongue, close your mouth, and see if you can taste it. If you can taste it, you are going to go to the third page here, and you're gonna write your phenotype. Your phenotype for me would be taster. Here's your choices, taster or non-taster. I'm gonna write I am a taster, okay? And somebody that can't taste anything funny and it would taste really bad, so you would know, you're gonna write non-taster. Then my genotypes, okay. This is where the front of the lab is really important. You need to read these as you go along. It says on number 3, your possible genotypes for your, what your genotypes could be. Tasters are either TT or, big T or big T, little t. So if you're a taster, you're going to write both of these, okay? Because you read the directions and you know it means you could be two things. So I am a taster and could be big T, or big T, little t. Okay, and if you're a non-taster, you're going to be little t. What does that mean? That means if you're a taster, you don't know if you have two dominant, that's why it's capitalized, you don't know if you have two dominant traits for tasting, or if you have one dominant trait and one recessive. Either way, it shows up the same way. The one dominant trait bullies and hides that little recessive trait in pure dominant and recessive. There's no blending. So this dominant trait overrides the little trait. Okay? And so you don't know if you're big T or big T, little t. Okay. You're going to go through all these other traits, and the description for like hairline about having a point in the front called the widow's peak or a straight hairline. All of those descriptions and what their genes are all listed all the way through this lab. And you're going to go through and do that whole lab, recording your traits all along the way. Down here, when you get to incompletely dominant traits, hair texture, eye distance, or lip protrusion, this is where we see a mixing of traits. So you could be any of these. So for me, hair texture, I have straight hair. But, so for straight hair, I'm going to look up what the genes are. And for hair, that is for straight hair, it's SS. So I'm going to write that down here. But for inner eye distance, I have a medium eye distance. They're not close, they're not far apart, they're just medium. Okay, so I'm going to write medium right there. And then for here, you'll see on the descriptions that for eye distance, for medium, that is a mixing of traits, two capitals, D and F. So what's important to note here is that some genes, all the way through here, these genes are all pure dominant or recessive. There is no blending of traits. But down here, there's some examples of traits that are mixed, that are blended together. So here you'll have some blending, and up here you will not. You'll answer all of these questions. One that kids get hung up on in home study is which of your traits you share with your classmates. And they say, well, I don't have any classmates. Well, go and check with your friends or your family or whoever is around. Look at their traits and see what do you have in common with them. What is totally different than what they have. What traits compared to your friends are unique to you? If you had anybody that you have all of the same traits, what still makes you unique? You can answer that question without even knowing anybody that has all your traits in common. Just if you had all the traits in common, what would still make you unique as a person? You need to read and find out in your book what determines your traits. What determines your traits? I can tell you that it has to do with your genotype, but you need to come up with a little more information than that. How can a person's genotype for a trait be determined from his or her phenotype for the trait? So your appearance, your phenotype, if all we can do is look at you, how can we kind of figure out what your genes are? Well, there's lots of ways you could do that, but you think of one, like if all we could do is look at you, how can we determine what your genes are? You spent a whole lab data table session doing that exact activity right here. So you just need to translate that into written form right here, what you did. And then number 9, why was untreated paper used in the PTC test? Why did they ask you to taste test regular paper before you taste tested the PTC paper? Why did I do that? This further exploration here you do not need to do, so you can skip that. All right, have fun with the determination of genotypes from Phenotypes Labs. You can learn a little bit more about yourself and your family and friends. Take care.